So we might get this last session going and see how we go with time to get you out in time. But look, the first three sessions have really been fantastic. I've really enjoyed them and the diversity of talks. This session is about prevention and so crucial in AMR and something that we really need to focus on in a big deal. And I think this session really also highlights the cross-disciplinary nature of this centre and also our efforts in AMR, where we've got such three diverse speakers, from an aerospace engineer to a clinician researcher to faculty of arts. So I think this is going to be really exciting. First speaker is Associate Professor Andrew Stewardson, who I'm delighted to say is our new Director of Infection Prevention and Hospital Epidemiology at Alfred Health, which has just been announced in the last week and a half. So we're very excited that Andrew has taken up that position. Also a clinician researcher, NHMRC ECF fellow, and has a big interest in infection prevention related to AMR. So welcome, Andrew, and look forward to your talk.
Thanks, Anton. Thanks, and thanks very much for the invitation to speak today. It's been, really enjoyed the day. So I'm going to introduce this talk by briefly kind of trying to place infection prevention into AMR and where it might fit. And then I kind of took the symposium name literally and thought that I would just present a few unmet needs in infection prevention with regard to AMR. So I think Ben said earlier on that it's bad form to present problems without solutions, and I'm sorry I fell into that pitfall, but I'm hoping that other people might have some solutions to offer. So this is Australia's national AMR strategy to 2020 and beyond, and one of the four pillars there in the middle is prevention and control of infection and spread of resistance, always mentioned in national strategies for antimicrobial resistance. And if you don't believe that, the government then, earlier on today, Adam Jenny said prevention is better than cure. So that's hopefully one of those two will convince you that it's important. This is my highly complex conceptual model of infection prevention and its role. So we have a person who has an infection, they get exposed to antimicrobials, which leads through multiple different mechanisms to colonization with antimicrobial resistance. And then there's two things that could happen. Firstly, transmission to other people, or infection themselves, moving from colonization to infection, which then again leads to more antimicrobial usage. And so what we're trying to do with infection prevention, and I'm particularly talking about the hospital healthcare environment during this brief talk, is try and prevent the infection to start with, try and prevent transmission from someone who's colonized, and then also try and prevent progression from colonization to infection. And so the next question, it might be, haven't we sorted out infection prevention? I think there's sometimes an assumption that, we have PPE, there's hand hygiene, there's sanitation. We sorted these things out a long time ago, but fortunately for the sake of this presentation, we have plenty of unmet needs. And so what I thought I might do is just use carbapenemase producing Enterobacteriales as an example, and think about four questions that I think are unmet needs with regards to infection prevention. So can we eradicate environmental reservoirs for CPE? And for each of these questions, I'm hoping it sort of highlights a group of themes that either are currently being applied to that question or should be potentially. So we're looking there at the built environment and also at engineering. Can we institute CPE infection prevention measures without negative impact on patients? And there it's more about implementation, sociology, consumer engagement. Third question is, can we ever clear CPE carriers, which is something we were just talking about in the break, in fact. And they're moving back to the microbiome and therapeutics. And then finally, can hospitals work better together to prevent CPE transmission? And they're touching on mathematical models, genomics, and policy. So to start with, can we eradicate environmental reservoirs of CPE? So this is a cluster of patients. So each line there is a patient, and the circle or the square or the diamond is the moment when they were first detected with CPE colonization. This is in the intensive care at the Alfred Hospital back in 2017, in fact. So about 20 to 30 patients around, clustering around a few months. And what we found was that as a potential source of that infection, without going into too much detail, they were all IMP4 carbapenemase producing enterobacteriales, and most, as highlighted here in red, of the unfortunately of the ICU sinks were also found to be contaminated with IMP4 producing enterobacteriales, in Serratia in particular at that time. So if you look at the literature, and it's pretty much the same now. If you go and look now, a few years later, there's lots of options that are discussed. There's to remove and replace the plumbing. Unfortunately, seems to just result in recolonization down the track. Chemical cleaning, steam cleaning, drain covers, self-cleaning drain traps that use heating and vibration. And here's one paper that reports the application of that sort of, sorry, this is acetic acid. So this is a group that used acetic acid to decontaminate sinks successfully and resulted in reduction in colonized sinks and patients. And this is another one that used the machine that you put underneath the sink, which applies heat and vibration and results again in reduced contamination and patient contamination. Patient colonization and infection in this particular case. We were actually, I remember we were talking about some of those things, but they all had barriers to access at the time, and our engineering department said we should use this instead. So Blue Thunder, it's phosphoric acid. It's used for multiple things in industry, and you also might be familiar with it as an ingredient of Coca-Cola. Which may be not surprising given some of the things that you can use Coke for as well. And so this was the intervention that was put in place in 2017. So in addition to some other things which I won't talk about relating to sink hygiene and faucet replacement, we poured 250 mils of this into the sink, put water on top with the plug in, left it there for a sort of dwell time, and then pulled the plug out to flush it through the system. And we did that 3 times weekly for an induction sort of phase, and then a maintenance period of once weekly. And each line here now, instead of patients, we're looking at sinks. Red is the contaminated sink, and sort of tealy kind of color is negative. It's not really, unfortunately, the induction and maintenance phases should be projecting, but aren't. Sorry about that. But basically, we showed with several different cohorts that we went from pretty much all sinks being colonized with CPE to clearing them out pretty quickly, which was terrific. But wasn't by any means I think the ultimate solution, because this process is pretty disruptive in the clinical environment. It's resource intensive. It doesn't eradicate, it just suppresses if you stop, which we found during COVID. It comes back again. And then there's also wear and tear on infrastructure. So it does damage to the infrastructure, as Denise and Megan, who are sitting here, are quite familiar with from having invested a lot of time. So that's why there's also a lot of interest in this sort of concept of just completely eradicating water from the intensive care unit, or having a waterless ICU. And so that's my brief touching on that first challenge of environmental reservoirs. Second challenge: can we institute infection prevention measures without harming patients? And this will be quite brief, but just to say that when we find a patient who's colonized with CPE, we put them in isolation precautions or contact precautions. So they put in a single room with their own ensuite bathroom, ideally. Staff wear gowns and gloves. Investigations and procedures. There's a cleaning requirement after they get transported out of their room to somewhere else to have something done, which often means that they're put last on the list, possibly sometimes even cancelled, or at least most likely of being cancelled. And there's risks associated with all these things, including being isolated, as we learnt from COVID, obviously, has an impact on mood. There's a barrier to communication. There's just that extra barrier of going in and talking to patients, and delays for all sorts of things, including just delivery of meals or diagnostic procedures and investigations. So there's a few systematic reviews. This one looked at patient harms, 26 independent studies, which reported a wide range of both psychological and non-psychological outcomes, and certainly a trend for higher levels of depression and anxiety, and also worse outcomes with a range of care-related factors, comparing people who are in isolation precautions to those who are not. These are not high-quality studies, so there is certainly a risk of some confounding there as well. Patient experience, this systematic review of 15 independent studies suggested that people in isolation precautions were more likely to report lower ratings with regard to their own experience of being treated with respect, having high quality communication, receiving assistance in their care, and also cleanliness. And then, often it's more meaningful to hear the patient's voice itself. So this was a paper that was done in a study in France, interviewing patients in general practice after they'd been in hospital and isolated with a multi-drug resistant organism. And you can see, it worries me, it scares me a lot. I don't want to live with a sword of Damocles hanging over my head. It's coming back all the time. And most patients really interested in wanting to be free of carriage. When can I be free? When can I stop worrying about this? And there's been other qualitative papers describing anxiety around contact with family members, and do I have to worry about having close contact with my grandchildren and so forth. So that leads on to the third challenge, which is can we ever clear a CPE carrier? And at the moment, the Victorian state guidelines for carbamazepine producing Enterobacteriales say that it's not possible to ever say that a patient who is colonized with CPE can be declared cleared. So they must always, isolation precautions must also always be implemented upon readmission of a case to a healthcare facility. We can't ever sort of do a certain number of negative tests and then say that patient's cleared and can be released from isolation. So that's not only the sort of contact precautions have the risk of harms, but it's sort of indefinite for the rest of that patient's experience with healthcare at the moment. So there's not a huge amount of research done prospectively looking at duration of colonization. There's a few. This is one here from Singapore, 21 CPE carriers only, followed up for about a year, showing that a lot of them no longer had colonization at a year. I think the estimated mean duration was 86 days. That was using a modeling sort of approach to account for interval measurement of colonization. And the probability of decolonization in 1 year was 98%. So most people could no longer detect CPE after about 12 months, but that duration was impacted by and prolonged by exposure to antibiotics. And unfortunately, we know that a lot of the patients who are colonized by CPE have have a lot of comorbidities and tend to be in and out of hospital and exposed to antibiotics again. So that probably does apply to quite a few of the patients that we see with CPE colonization. So can we actively decolonize CPE the way that we do with other antimicrobial resistant drugs like bacteriocin, like MRSA, where decolonization is a known strategy? This was a study led from Geneva, and people with an interest in the microbiome might want to avert their eyes for the next few minutes. So this was, I think, the first blinded randomized trial looking at decolonization, ESBL here, not CPE, extended spectrum beta-lactamase producing Enterobacteriaceae, and they used non-absorbable antibiotics, colistin and eomycin, for 10 days. And if patients had urinary colonization with that organism, they also had nitrofurantoin for 5 days. And so what they found was, so we've got the fraction of rectal ESBL carriage. So after 6 days of treatment, you can see a big difference down to just 33% or so of the intervention arm being colonized. Also a fall in the control arm as well. And day one, post the completion of that 10 days of therapy, that same sort of trend being seen, but unfortunately, a week after completing therapy, back to sort of the same sort of level, and that trend sort of continued on there. So it's really just suppression, again, that sort of concept, a bit like the sinks, so suppression and then rebound. So the same group then went on to think, what about decolonization followed by FMT, or sort of eradicating and then replacing it with something that's a bit more benign. And so they did this incredibly complicated study, which was really hard to recruit patients to, where patients had the same regime of colistin or neomycin, but then either had fecal microbiota transplant or a capsule formulation, depending on which study site they were in, to try and stop that rebound after the withdrawal of antibiotics. And I can't remember if I decided or not about whether to highlight this or not. Yeah, there I did. Okay, so that actually I just wanted to point out that in this figure, they've got a key here. It says brown circle, stool culture obtained, which I don't know. Anyway, maybe it's just me. And a pretty similar result. So a bit of a difference during the antibiotic phase, but then rebound. And unfortunately, they weren't able to recruit the number of patients they wanted to recruit. There was no significant difference. So that's, I think, still an open question about decolonization for multi-drug resistant gram-negatives. And then the final challenge I wanted to mention was, can hospitals work better together to prevent CPE transmission? Hospitals, I guess it is a bias. Working in infection prevention in hospitals, I think that hospitals are a focus for amplification and dissemination of antimicrobial resistance. Traditionally, infection prevention units have worked independently within their own hospitals. They haven't coordinated within a system, and so everyone considers themselves their own island, epidemiological island. But there's sort of an increasing recognition that surveillance and containment actions should be based around the network sort of approach and everyone should be working together more efficiently to try and develop interventions. This is not actually a new idea. So here's a paper from 2005 which used a modeling approach. Their hypothesis was that if you apply a game theory lens to infection prevention, then there's a benefit for hospitals to not be so proactive about spending money on infection prevention and just benefiting from other hospitals in their own network who are spending money and working on infection prevention. They modeled to suggest that will always be a problem unless you have some sort of regional coordination for infection prevention. This was data-free sort of modeling. More recently, over the last 10 years or so, there's been use of large data sets of hospital admissions, nationwide data sets of hospital admissions, sometimes linked with genomic data, looking at the spread of multi-drug resistant organisms within countries and between hospitals. And so these are two studies from the Netherlands and from the UK, looking at each point there is a hospital, and the lines between the hospitals, the edges between the hospitals, are links, the strength of which is determined by how many patients are transferred between them or readmitted from one hospital to another. So it's thinking about hospitals as networks, and the fact that they cluster together, and not every hospital is in a similar spot on the network, and your position on the network is likely to influence the prevalence of antimicrobial resistance in that hospital. And so there's a few things that you can do once you've got that sort of network set up. And the first is you can link it with the genomics and use that to try and to test the hypothesis that the shared patient network translates to differing risk of spread of multi-drug resistant organisms. And these are two papers that show that actually is the case, that you can explain how antimicrobial resistance transmits around a country by using hospital networks. And then you can also design an efficient sentinel surveillance system. So maybe you're wanting to look for the emergence of a novel antimicrobial resistance, maybe Candida auris or something else? Do you need to test patients in every hospital, or could you just pick the ones that are at certain places in the networks to make it a sort of efficient approach? And so that's what this paper here did. And then finally, using the model to design a cost-effective, efficient approach to implementing infection prevention interventions, and that's what this paper here did, was showing that even if you look at the CPE prevalence here, you can see that with 100% implementation of standard precautions amongst all hospitals wasn't quite as good as a coordinated approach to CRE, which might not actually be as resource intensive as the 100% implementation. And just to flag that this is something that we are trying to start to look at with a project, an NHMRC funded project at Monash. And a lot of work went by Michael Liddymore, whose photo's in the corner, to produce this simple looking figure, which is the network representation of hospitals in Victoria. So the circle there is all Victorian hospitals, and the outer ring is private versus public hospitals, and the inner ring is a clustering of hospitals depending on connectivity of shared patients. And we're trying to use this to do some of that work that I mentioned earlier on about designing efficient interventions and a sentinel surveillance network, for example. So I think that's, so here we're wrapping up. So there's no shortage of unmet needs regarding antimicrobial resistance in infection prevention. Just some examples that I tried to present here were the need for cost-effective methods to control AMR environmental reservoirs, particularly sinks, an approach to AMR case management that doesn't harm patients, better understanding about the duration of colonisation and methods to eradicate AMR colonisation, and health system level strategies to detect and respond to AMR rather than every hospital being its own independent agent. And acknowledge all the people that I work with. Thanks very much.
Fantastic. Thank you, Andrew. Awesome talk. We might just take one question and then we'll leave some time at the end. So is there a question at all from the audience to Andrew? Yes, Carl. Oh, and then we're going to go to Megan after Carl. So we're going to take two questions.
Great talk, Andrew. So it's all well and good when you know they're colonised, but there's always the bottom of the iceberg that you can't see. How do you build that into such structures so that when the patients you can't see that have CP or you haven't tested yet, and then pushing that colonisation through your institution?
Thank you. I feel like it's a planted question, because one of the other unmet needs I was going to put in there was the tension between horizontal and vertical infection prevention measures. And so, as much as we can, trying to move away from targeted sort of vertical measures, where you need to have the detection, and back towards horizontal measures, which are sort of universally applied. I think that's a much more resilient, robust sort of approach. Absolutely. And I suppose you can think about that both at the hospital level, but then also at the system level. And that's one of the things that we want to try and look at with that last project that I mentioned, thinking about how do you design a health system to try and minimize the spread of antimicrobial resistance. Not that that's going to be the only thing that feeds into how you design a health system, but maybe we can provide some advice. But thank you. That's a really good point.
Thank you.
Did you come up with the Echidna or did Michael?
Actually, that was. I do feel like I can sort of retire at some point happy having come up with that acronym.
Because you've been questionable with naming things before.
Thank you. Yeah.
Great. Thank you, Andrew. Hands together for Andrew. Thanks. All right, we'll move to our next speaker, which is Victor Cardasso. So Victor is from the Mechanical and Aerospace Engineering Department at Monash University, and is really doing some exciting research and really some applications in the context of pathogens and AMR. And we really look forward to hearing your talk, Victor. So thank you.
Thank you. Thank you, Anton, and thank you for inviting me today. Let's see if this works. Perfect. So, Anton mentioned I'm an engineer, so I apologize. But we are also in this space, and I'm really happy that you're welcoming us, because I think like the way that we are going to go through this and the way to success is when we work all together into this to find the solutions. What I'm going to be talking today is how we can use micro and nano topographies on biological applications. And what I mean here is basically for the last few years, we have been working on implementing small topographies on top of surfaces that we can use to influence the behavior of cells. And those cells can be mammalian or can be bacterial. And the combination of those things, we believe that will have a massive impact when we put it on implants, so that we can both reduce or at least reduce the risk of infections, but we can also have other active effects on those implants. And I will show you one example that is thought about towards orthopedic implants today. So all of this is based into something called mechanotransduction that many of you probably know, in which basically cells are extremely sensitive from mechanical cues in the microenvironment. The cells are sensitive to chemical cues and so forth, but mechanical cues have a huge impact. And those cues can come in many forms. They are very sensitive, for example, to the stiffness of the surfaces where they are. They are sensitive to stresses, shear stresses. They're sensitive to changes in pressure, and they're sensitive even to acoustic forces around them. But they're also sensitive when there are topographies on the surface. And those topographies is what we can engineer on the surface, so we can elucidate very, very specific response from those cells. And this is what we are trying to do here. So first of all, I'm going to be talking on how we can use this to, for example, direct stem cell behavior for mammalian cells. And this is a collaborative work. The work was done mostly by two postdocs, James and Hassem. This is a collaborative work that I did at Monash together with Nico Volker and Jessica Fritz. And there's a whole range of other collaborators from CSIRO, Peter Mack, EPFL, and the Max Planck Institute. So here what we did, among other things, and I'm not going to go into all the details, but we wanted to create a technology that allows us to investigate those topographies very efficiently. So we created was this technology that allows us to implement these topographies on tissue culture plates. And we wanted to do that because, as you know, tissue culture plates or like well plates are like the workhorse on cell biology. We wanted to have something that was fully compatible for biologists to use and to have high screening capabilities. So we created this technology that allows us to create all these micro nanostructures with different types of shapes, different types of topographies, with all the different configurations that we wanted, so we could do these studies very, very efficiently. What this allows us to do is this high throughput screening, and then we tested it against a whole range of different mammalian cells here, including MSCs, fibroblasts, etc. And what we realized is that thanks to this technology, we could start to identify cell phenotypes very, very quickly with automatized microscopy and so forth. And by these techniques, we could identify those phenotypes that have very specific response, like, for example, a determination of fate onto this stem, for example, on stem cells. And here what we could do is identify some particular topographies that had an increase in osteogenesis. Now, once we understood that that was happening, we wanted to understand why this was happening. And what we did was look into these cells specifically. We started looking into those topographies with a lot of care. And then what happens is the following. When you have your cells on flat surfaces, they look fairly normal, as you will imagine. But in the moment you put them on top of those topographies, obviously the morphology of the cells change radically. And that's something that obviously you can identify very quickly when you look in things like the cell area. And you can see that the cells there, like they exhibit like they have some holes and stuff. Those are not really holes, and we will talk about that in a minute. But what was more interesting for us is that you're not only changing the morphological phenotypes, you're also changing things like, for example, protein expression and gene expression by those cells. And those are the things that we are really, really interested in, because those are the things that are going to have a real impact on the cell behavior. So if you look now into details into those cells, because we wanted to understand why those changes were happening, what you can see is when you look at the nuclei of those cells is that they have shapes that are quite dramatically deformed. Like in this case, they almost have like a donut shape with a hole in the middle into the chromatin. When we saw this type of cells, the first thing we thought was, okay, those cells are not going to be very happy with those type of deformations. But then we started checking that hypothesis and we were completely wrong. Those cells were really happy, they were proliferating. There was absolutely no reason in any of the assays that we tried that indicated that those cells were not actually enjoying themselves. And as a matter of fact, when we started checking how this was actually happening, what we identified was that the cells were doing this themselves, like they were pushing themselves into those structures and they were forming those deformations by their own force. In the moment that we applied some drugs that prevented the cytoskeleton to produce stress forces, those deformations disappear. So it's the cells themselves that were pushing themselves into the structures. Now, what happens here is that the nuclei is actually not perforated. The DNA around the nuclei or in the nuclei is deformed around the pillars, but the membrane is intact. The membrane has not been broken at any point. What was very interesting here is that when we had this type of deformations, and very specifically this type of deformations where there was that inner perturbations, where you have this type of holes in the nuclei, what we observed is that that was the ones that was giving us a highest regulation on osteogenesis. So when we had that type of deformation, we had the increase on osteogenesis from these cells of close to 4 times, which is very, very significant when we are not adding any additional drugs or anything. This is only the effect of those topographies on the cells. So we wanted to go beyond. We identified that this was because, thanks to these deformations, we are changing the localization of JAB, which is a protein that regulates osteogenesis. I'm not going to go into details for time's sake because I know we are time pressured, and I want to make sure we have some time for Mark after me. So. Obviously, all of this was done in vitro, but we wanted to check that this type of effects could be translated in vivo. So we were working with Michael Martino in Army, who has a mouse model in which they do these cranial defects in the mouses. And we tried some of those topographies to see if, thanks to the topographies that we have identified in vitro to have this type of effects, we could actually observe similar effects in vivo. And if you look at the graph down there on the right, the one that says that was pro-osteo was working worse than the ones that says that was anti-osteo. So we had completely opposite effect in vivo than what we have observed in vitro. We are at the moment writing a paper on this, and we have identified the reasons on why this was happening. And it's because basically these topographies are changing, obviously, the nuclei, as I showed you before, but they're also changing how the cells are colonizing the surface. And when we had some of these topographies, actually the cells were colonizing the surfaces much faster than if there was no topographies at all. The topographies that was giving us a highest osteogenesis was reducing the speed of colonization of the surfaces. And for that reason, we could observe that for this type of defects, actually the ones that in vitro were working worse was actually working better because it was speeding up the colonization of the surface that then helped to close the bound. And that brings me to the second thing that I want to talk, and it's obviously, here I'm talking about mammalian cells, I'm talking about what we can do with these topographies on mammalian cells, but this impact of topographies and colonization has been also shown in bacterial cells. And this is the second thing that I want to discuss today, because that's a work that we did with Trevor and Ian Hay, who is now in New Zealand, and my student Sarah was doing this work. And here what we were looking is, okay, we have this type of topographies, and we expose, like we try to colonize these topographies with bacteria. Can we deter the colonization, and can we deter the formation of biofilms thanks to the presence of topographies? Because if we can do this, and we can think about an implant in which we can have some sections, for example, that we're promoting osteogenesis, so that you will have an improved bone integration, And in the areas where you don't need bone, you can put topographies that are going to prevent bacteria. You may have a strong impact on reductions of infections for this type of clinical interventions. I'm not going to go into details here. You have seen this morning the talk from Helmut, in which he was already presenting a lot of work that has been done on for example, on bactericidals, very inspired from the wings of insects that he was mentioning. There was this really, really nice work from Elena Ivanova, who is now in RMIT, which they were using these micro, very sharp structures that was killing bacteria very, very efficiently, but was not affecting at all mammalian cells. There are examples, other examples inspired in nature from lotus leaves, from shark skin. All of these have demonstrated that we can indeed either kill bacteria or we can affect the formation of biofilms. But there is one limitation, and is that when we think about applying this type of technologies in the hospital, in reality, we have to deal not with one type of a microbe, but we have a whole range of different things happening at the same time. And that makes it very, very difficult to find one solution that can work for all of them. That's why using technologies that help screening, as the ones that I presented before, can be extremely useful to identify some of these topographies that can be effective against multiple microbes at once. So the first thing that we did was create this range of different topographies. And again, for the sake of time, I'm not going to go into the details on how we came with all these different types of designs, but if you're interested, I'm really happy to talk afterwards about it. But the idea here was to, in a very simplistic way, make the life of the bacteria trying to colonize the surfaces very difficult in terms of finding a spot where they will be happy for doing this colonization, but also making it difficult for them to find each other so that they cannot start forming microclusters that will lead later for the formation of a biofilm. Now, what happens is when we use these micro nanotechnologies, typically we have topographies that look like this, that you have relatively sharp edges with vertical sidewalls. And this is the standard thing that you will obtain when you fabricate this type of micro nanotopographies. Now, what happens when you do that is that if you have a flat substrate and you put some E. coli and you wait 24 hours, what happens is that those E. coli start forming this sort of microcolonies that later on will lead to that formation of a biofilm. If instead of a flat substrate, you have topographies as these ones that I'm showing here with this sharp pattern, as we call them, you don't have those microcolonies anymore, but you still have lots of bacteria because they love those small holes that you have created. They like to hide in there. And effectively, in many cases, depending on the concrete design you are using, you end having more bacteria than what you have originally. Because even if they are not forming those microclusters, they are still loving that surface and you have lots of bacteria in there. So that was a problem for us because maybe you are deterring the formation of a biofilm, but if you have an implant that is at the end full of bacteria, that could lead to an infection later on, and that can be extremely problematic. So to prevent that, we came up with the idea like, what will happen if instead of having this type of sharp patterns, we create something that is diffuse, it's something that is curved, that is having this type of curvatures that is actually going to help both to prevent the bacteria from adhering into the surface, but at the same time, it's going to make that surface much easier to clean. What happened when we did that was that we moved into the third stage, in which actually you are reducing both the formation of those microcolonies. You can still see some of them depending on the design that you have, but they are much smaller. But overall, you're reducing significantly the number of bacteria. And all of this was done with E. coli here. So what happens is like when we use this type of technologies, and again, I'm not going into the details on how we fabricate those, but if you're interested, please come and talk to me, and I will talk to you for hours if needed. Okay. So what happens, like they basically can be implemented in this type of fashion that you have these smooth type of topographies with this added implementation. So we tested it with 3 different bacteria, E. coli, Klebsiella, and Pseudomonas. And then what happens, like when you had the flat one, they look like this, where you have, in all the cases, after 24 hours, you have these large microcolonies starting to form that will lead to problems. If you have the structures as the initial ones I showed you with that sharp configuration, which is the classical one, what happens is like as I said, you in many cases are reducing dramatically the number of microcolonies for pretty much all the designs that you can think of. But in terms of bacteria, the total number of bacteria either is not really reduced or it's actually increased in some cases. Like here for Klebsiella, the number of bacteria was just going over the roof when you had this type of configuration. But when we implemented the diffuse designs with this type of 3D topographies, we could obtain both things simultaneously. We were reducing the number of microcolonies, and we were reducing significantly the total number of bacteria on those surfaces. So in our opinion, that was a much better outcome in the whole thing. Something that I should highlight here is that here we selected one of these multiple designs that we created, because this design was working fairly well for the 3 bacteria simultaneously, but it was not the best design for either of them. There was other designs that was much better, for example, for E. coli, that was much better for Klebsiella, or much better for Pseudomonas, but those were not working as well for the other bacteria. So having this type of studies where you are actually analyzing multiple microbes simultaneously, which they have different methods of colonization of the surface, is extremely important, because otherwise you're losing that critical information that is going to be indicating what type of solutions will be better for a more generic approach. And with that, I would like just to highlight a couple of things to end. So I hope I convinced you that the use of these surface topographies can have a massive impact both for mammalian and bacterial cells, that it is possible to produce effects that are complementary and that we can integrate them simultaneously when needed. We need to develop technologies that allow us to scale these things up. And again, I haven't gone into the details of technology because of limitation on time and because I think it will be more interesting for you to see the actual effects and applications. But all these technologies we have implemented here are actually easy to escalate and easy to implement into a large scale, large areas, and so forth. What we are working at the moment is to translate this into actual 3D objects like implants and so forth, which the technologies, we don't have them today, but they need to be developed, and that's part of the work that we are doing. So thank you for your attention, and thank you for the collaborators and my group.
Fantastic, Victor. Question. One question before we... Yes, Simon.
I was just wondering, in terms of the range of surfaces that you can coat, you mentioned implants there at the end, but I mean, what sort of scale do you think you can achieve? I mean, you know, if you can have an effect on biofilm formation, you know, can you roll these surfaces out onto furniture and benches and around the sinks and all that sort of stuff?
My dream will be that we can put the surfaces at the bottom of large carrier boats. Are we going to do that today? No. Are we going to do that in 10 years? Maybe not. But I think it is possible. As I mentioned, like all these structures that I showed you today, those are fabricated with a standard UV lithography. So you can imagine that you can pattern like really large areas with them. And certainly into furniture, into small objects like handholds, buttons in elevators in a hospital to prevent cross-contamination. These type of things are very low-hanging fruits. And those are some of the things that we are aiming to do in the near future. Implants are very interesting from obvious reasons because of the immediate clinical impact that they will have, but just general surfaces in hospital, in areas of high risk, areas of high contact, in public transport, that we can reduce the amount of disinfectants, for example, you need to use to clean them up. All of those things will have a massive impact. So yes, like all these technologies are aimed to be able to be compatible to that, but they are being developed, like they don't exist today.
Fantastic. Thanks, Victor. We might move on and we'll get you back in the panel. Okay, yeah, excellent. All right, so our last speaker of the day. So Associate Professor Mark Davis, who is from the Faculty of Arts and always provides us with really insightful perspective on the ways we communicate a really challenging topic, even the use of the word AMR and superbugs and things like that. So Mark always gives us really important insights. He's going to teach us the ways of how we need to communicate and get the message out And I look forward to your talk again today.
Thank you. That's very kind. How do I find myself here? Yeah, thank you so much for inviting me to talk. My colleagues in the centre are so careful to include the social scientists. So it's really great to be here and to have listened to all these wonderful talks. So what I'm going to do is talk a little bit about some work that was kindly funded by the centre actually to systematise our conceptualisation of how to prevent, how to educate and communicate around the AMR crisis in the general population. So I don't have any experiments or kind of imaging to show you of cells. I'm just going to talk about people and the world out there. I want to talk a little bit about the effectiveness of social marketing and how that might frame how we might move forward. So a bit of an audit of the social science there. Talk a little bit about the social and cultural factors that mediate how individuals in our world think about bodies and infections and immunity and superbugs. Summarise a little bit of the work that went into this document around the kind of program insights. So looking at the grey literature published by the international agencies, our own work around systematic reviews of evaluations of effectiveness of different kinds of social marketing campaigns, and the kind of insights generated out of that work. And then I want to propose how we summarise the work in this document, which talks about something called integrated AMR prevention. So I'm going to offer a kind of way of conceptualising how we would do prevention that draws together some practical kind of ways of doing things. And I also promised to talk about COVID. So I think that's a bit silly, really. So I'll make some passing comments at the very end if I have time to talk about the impact of COVID on how we're doing AMR prevention. So the policy context. So many of you will be familiar with the One Health Master Plan, which is very explicit about whole of society orientation to the crisis. And so there's from the top down, there's a kind of an approach that we should be including general publics in the kind of move to prevent the superbugs crisis and minimise it. So I'm kind of levering off some of those ideas and developing them. So what do we know about general populations and the sort of things that are happening in the world out there to let them know about superbugs and to alter their behaviour, their kind of demand for antibiotics, their trust of advice not to have antibiotics or to use them effectively, to support efforts to generally respond to the crisis. So we've been doing this at least since 2000. That's as far back as I've been able to go to find real world examples to actually look at. And we're finding that, there are issues. Public awareness doesn't work so well. So, for example, LUM found that when they spoke to uni students, none of them could remember any public awareness campaigns. They knew about superbugs, but they couldn't kind of generate spontaneously insights about what was happening out there in the world. Evaluations of Antibiotic Guardian, the pledge website. Have you all pledged? Yes, thank you. Evaluations show that these work really well, but the people who are pledging are us, okay, not the general population. So scientists, healthcare professionals, the converted basically. Same in the Twitter sphere. The content analysis and the network analysis of how that's working. That really is a kind of a technology that reaches all of us, but doesn't penetrate much further into the world out there. If only Trump had a superbug. Did I say that? Oh dear. That might work. We've also done our own awareness, looking at the kind of social, mass media actually, television, newspaper and online news in Australia. And this is a kind of glimpse for 2017 about the kind of actually just counting things, news items about antimicrobial resistance. And I think this graph basically says there's no pattern. There's no relationship between what we might be doing in November for Antibiotics Awareness Week and the response in the kind of media environment. So we're kind of in a phase of communicating about superbugs that's at the tail end of what was established really in the early 2000s around golden staph in hospitals in the UK. That was the peak time for news items about antimicrobial resistance, and we're kind of in the kind of failed or the zombie zone for news items. When we ask people what they know about antimicrobials, small majorities know the difference between bacteria and viruses. And when we sort of survey people who are coming out of hospitals about infections, most of them fail to understand what antimicrobial resistance is. So the picture here is, the messaging is not really cutting through. The thing we need to bear in mind too is that most of us in Australia access oral antibiotics on script. It is possible to do it otherwise, but most of us are good citizens. And that prescribers are offering antibiotics to kind of reinforce their relationships with their patients. They're worried about, preventing worse illness in vulnerable populations. They're prescribing due to time constraints, and they're thinking about their businesses. So another dimension here is that really we're not living in an environment in Australia where access to oral antibiotics is kind of just a free-wheeling kind of environment. The clinical encounter is key to the distribution of antibiotics into the population. We also know a lot about how people think about these issues. So when we've interviewed and done focus groups with all kinds of people from the general population about what they think is going on, they all endorse the principle of the need for AMR prevention, but they're not really aware. Some of them don't even know what an antibiotic is. So you might ask them, have you had an antibiotic? But they could get that wrong. So this, the kind of awareness and knowledge and insight into what's actually being taken into their bodies is kind of diffuse. People who had higher levels of education and an interest in science were much more able to describe antimicrobial resistance and kind of produce an orthodox account. If people weren't highly educated and or not interested in science, their grip on AMR was kind of flimsy. Another important thing to think about is a kind of culture around the body and how we understand our bodies as the site for our health. So repeatedly in the qualitative research, members of the general population talk about resistance as my body is resistant. Okay. So, and you can correct that knowledge, but they'll revert back into that idea that the body is the fortress and they're protecting it, and that's how they think about their kind of worlds. Just as an aside, so this is doing a Google Trend search on immunity, the word, worldwide, and this little bump is, guess when? The beginning of the COVID pandemic. So I was kind of fascinated that whatever Google Trends measures, there's this kind of interesting lift in searching. It can't just be me. Globally, people started to think about their immunity as the way to respond to this emerging threat. So, meeting people where they are at in terms of how they think about their bodies is kind of a crucial kind of dimension here. The other thing to kind of patch into, if we're trying to conceptualize how we ought to kind of program for prevention in the general population, is how we message. So I want to get you thinking about polysemia. So we produce a message. It doesn't always have one meaning, and it can be read differently by different people in different ways, and also transit over time and be reformulated. So there's a few things to kind of factor in here. It's a bit of a kind of, rag bag of kind of principles of communication that I have used to think about why we need to do integrated education. So the antibiotic apocalypse narrative, it's probably going to produce countervailing effects. So threat messages, fear, we know that works temporarily, but people habituate to fear messages, so you need to escalate the scariness. And we've been through that in Australia. We had Grim Reaper, and that had a kind of impact that kind of released other demons as we did that. Also, threat, frighten people. You need to tell them what to do to avoid the threat. That's really kind of fundamental. You could just scare people and they don't know what to do. They're going to stop listening. We also need to think ethically about fear messages. Are they sustainable? Is it right to add to the psychological pressures of prescribers and people who are ill and consuming antibiotics, etc., and so forth? The other thing about apocalypse messaging is that not everyone responds as a good citizen in the face of scarcities. That's why I've got the supermarket image. Some people will say, well, give me the antibiotics before they're gone. I want my share packed away in my cupboard just in case. So these things need to be considered when we formulate strategies. Another favoured kind of approach coming through is behavioural economics. And we need to think a bit about that as well. So this is the idea that you bypass the sentient being and nudge them into behavioural responses. I wonder about trust and whether behavioural economics actually encourages relationships with citizens that are democratic and deliberative. And if we don't actually bring people with us, where do we end up in terms of public trust when things change, as they often do? So those are the sort of kind of cultural kind of dimensions. And then we're looking at what the evaluations are telling us about what seems to work. I could have a conversation about the science of evaluation in this field, and it's a little bit flimsy. So that's another whole kind of discussion, really. But for what we can tell, these are the kind of things that seem to be associated with good outcomes in social marketing. So multi-level approaches. I mean, this is kind of health promotion 101, really. Both, multi-pronged education to bring clinicians and their patients along. Global and local campaigns, you get the tiering effect and consistency, the same kind of message being reinforced at different levels. When you design a campaign, don't just focus on either the prescriber or the patient, keep them together. Just why we're actually doing focus groups now, bringing vets, GPs and consumers together to talk about solutions around messaging. And the final thing is to kind of engage health systems. So, the evidence seems to be pointing to the need to integrate what we're doing. So this is the sort of framework we came up with in the document. And I tried to make it a hierarchy, and I'm not sure it actually is, but maybe this helps you to remember it. So if we think, starting from the base of our prevention education, has to be governance. So we situate social marketing and link it with the other aspects of prevention that we know can help. We need to build the research base. The evaluation and the community participation research is a bit weak. We don't have any way of monitoring how the general population in Australia thinks and feels about antibiotics. There's no longitudinal data by which we could assess how things are changing in the social environment and whether or not any of our education is actually working. We need to think about the health system drivers of AMR, and I think that patches in with a lot of the conversations we've already had today about treatment and prevention, linking prescribers and consumers together always in education, assisting media experts, communicators, teachers, and the general population to be more reflexive with the stories we tell about AMR. What's wrong with the apocalypse kind of notion, what it does wrong. Mobilizing a civil society, acknowledging and working with social culture, creating kind of guidelines around prescribing that are specific to different kinds of settings, infections, populations, and offering individuals, general population members, things they can do that are practical. So I think, if this is an unmet need, is to kind of integrate our approach to prevention education in this kind of way. How am I doing for time? Can I talk about COVID? So just a few, 15 seconds. That's about all I'm prepared to talk about COVID, actually. So I did say I would talk about that, and we have got data from prescribers and consumers about the impact of the pandemic, because that's when we did our research. COVID was there. So, the legacy of the pandemic, are we moving into an environment where there's a kind of opportunity here for building a kind of social response, to like a preparedness for infections? I think we need to be careful about that, because maybe people are kind of over pandemic speak. How we message is kind of difficult. We noticed really strongly how much COVID disrupted the AMR workforce. Every expert we spoke with, every general practitioner, every vet, their work was kind of, decentered by the COVID pandemic. So if we're going to have a sustainable approach to AMR, we need to plan for that. We can't just, in 5 years, have another pandemic and stop thinking about AMR until it's over. Well, we can, but I wonder about the impact on our programming. And another little thing, and we're kind of working on this, it wasn't all bad, COVID. So the shift into telehealth in primary care meant that the doctors were paid to follow up with their patients. So if they prescribed an antibiotic, they could actually say, I'll call you in a week and see how you're going. That normally doesn't happen in primary care. There's not much follow-up unless the patient wants to come back. So it's kind of interesting how both patients and providers think about actually using telemedicine might be a way of refining how we prescribe and monitor the impact of prescription out there in the community. So those are my thoughts on education and also COVID.
Okay, if we're allowed to go 5 minutes over, which I think is pretty impressive for a full day, we could get the speakers to come up and sit in the panel. We might open up for some questions and discussion around the prevention of AMR piece. Yes. Oh, yep.
I work in fungal science, and after the release of Last of Us, I got flooded with emails from my friends. Are we going to be turned into zombies by fungi? So I think there was actually a lot of scope there, and also a lot of articles in newspapers were published about, fungal species threatening healthcare systems. So I think sometimes going drastic and playing with fear is actually a good thing, because I think it brings out like it has the potential to actually raise awareness of antimicrobial resistance. But I agree also that we have to add on to this, and then educate the basically the general audience in what to do with these informations that are now available through these TV series. So how do you think you add on to something, when there is a base, when people are jumping on this hype of, zombie fungi, where do you follow up on that?
Yeah, Monday evenings are ruined now that the program's finished. So there's a whole kind of genre around pandemics and entertainment. So, and there are multiple ways of engaging with fear. Some people find it entertaining. So, how you then program on the back of life of, The Last of Us is an open question. But I'll come back to the sector's capacity. If we had a AMR communications secretariat, full-time people who could kind of respond and work, use that media moment to build messages as part of an integrated approach, I think that would be really, really useful.
Great. And Mike, recently there was a tour around Australia about the documentary Salt in My Soul, which was about a story of a young woman with cystic fibrosis. What do you think the impact of that is, into a real personal story, a personal approach and insights? What do you think that approach is to engage with the community?
Yeah, look, really, really important. So cystic fibrosis, yeah.
Yeah, I mean, I think cystic fibrosis was her underlying disease, but more just a terrible scenario of eventual early death as a consequence of an AMR bacteria.
Yeah, look,
told in a very personal way.
Yeah. I think if we can tell people what they can do, the simple things they can do to protect themselves from threat, we're going to have more impact. We've used those stories for the longest time. They don't just exist in real life, they're also in the media. And people still don't know about AMR, and they hold on to this notion of the body as the kind of fortress. So, how do we work with that basic cultural form? And the sort of people who consume that media, they're the people who already know what AMR is.
Yeah.
All of those things.
And Andrew, do we know what to tell the community to do to prevent AMR? So there's the threat of it, and then what messaging do we tell them that what they can do to prevent AMR?
I think we tell people to use antibiotics, to not use antibiotics when they don't need them. But it's hard, isn't it, to operationalize that when you actually feel unwell, and weighing up the offer or the lure of an antibiotic that might potentially make you feel better versus seemingly intangible sort of esoteric risks that at some point in the future that don't seem quite so personal. So I think if you don't mind me bringing it back a little bit more towards infection prevention in the hospital, I think we're not very good at communicating about antimicrobial resistance and healthcare associated infections in the hospital at all. That's definitely something that we need a program for communicating about. Some at the Alfred have looked into that recently and found that people aren't understanding what we're telling at all about that. So definitely a lot of room for improvement in communication there.
I think that's where some of the other technologies that were talked about today and the diagnostics can really, you know, the better we can differentiate what someone in front of us has, the more likely we're going to be targeted and use an antibiotic in the right setting as opposed to the wrong setting. So, Kat, did you have a quick, was that a hand or?
That was a hand.
Oh,
Wasn't
yeah.
a wave. Although I'm very happy to wave at you, Anton. Mark, I so wish we'd had this symposium a week or two earlier, because this is so pertinent to a massive issue going on at the moment, which is the issue of doxy-PEP for STIs. And it's a really challenging issue. It's a very hot debate. Yeah, well, I feel like I'm terrified of giving a background, but yeah. So the background is that the use of doxycycline, either as a daily tablet or 2 tablets within 72 hours of sex, has been shown in a couple of studies to reduce the incidence of both chlamydia and syphilis, with some benefit for gonorrhea in some populations that don't already have really high levels of tetracycline resistance. So what we have is an epidemic of STIs in Australia, and particularly very high rates in gay and bisexual men, and very high rates of antibiotic use to treat those STIs already as a background. So methods to reduce STIs are important. It's just very unfortunate that the method that is being proposed and that has come up with is an antibiotic. So it involves more use of an antibiotic. I think the big question is, if you use that, does it reduce your overall antibiotic consumption? So are you actually at a net benefit? How do you control the use of it so that everybody doesn't end up on it and people are already buying it off the net, off script? And I meant just absolutely everything you said was pertinent to the 7-hour meeting last Friday. And the lack of knowledge of AMR, I think, was the most confronting for me, but also the fact that it was considered completely unacceptable for a clinician to place the burden on a patient about discussing or even mentioning AMR in a consultation, because that wasn't the responsibility of that patient. And I think that was really interesting for a clinician who actually discusses AMR routinely when I dispense any antibiotic or when we have an untreatable STI, which we already have. I routinely do it anyway. And you can do it in a sensitive, appropriate way, but it was actually considered quite unacceptable to put that burden on people. And the scare campaign stuff was brought up. The issue of what's right for me versus what's right for the community is it is a very, very difficult situation, and working in that space would be really helpful because you have so much knowledge.
It's on my list. I'd like an ARC to work with communities and clinicians around gonorrhea and so on. And you know, Duisberg, his theory about HIV was that gay men had overused doxy in the 80s prophylactically and it injured their immune systems, and that, you know, his alternative explanation. So doxy and gay sex has a long history and a politicized history. So there's scope for close focus research. I'm with you on that, definitely.
Any other comments, questions? Yep. Fish farms?
There's been a recent case of a couple of tonnes have been thrown in the water just out of Hobart.
Yeah.
It was only picked up through monitoring of wild catch.
Yeah.
I can hear the comment about that.
Yeah, I guess.
Was it just thrown out, or were they using it in the fish farms?
Yeah, I think
Doxy. I've not heard of that. Amoxicillin, I think, is what they typically use. I couldn't comment on fish farming. Sorry.
Anyway, it's a whole big topic, though, isn't it? Use in animal production and agriculture. Yep. Nana, did you have a question? Sorry, or was that all good? Yep. Victor, I just have one question for you. In plants, we are concerned by any bacterial cell attaching, like 10 or 100, any. So that design still has some bacteria attaching. So have you thought about that? Do the waves need to be tighter so it can't get into the grooves, or like?
Two things on that. Like, one, yes, we could further optimize all of these. Like, these are like pretty much the first kind of results we have been obtaining on that space. But also something to keep in mind is to do this analysis. We were colonizing this with really large amounts of bacteria. Like, we were putting thousands per milliliter of bacteria during the colonization step.
Right.
While in an accidental contamination, we will expect it will be a much lower dose. So effectively, it will scale down. So it is always a relative event that will happen.
Yep.
So, but by all means, until we don't try those things on in vivo studies with an animal model, eventually into clinical studies, but we will not fully understand what does the effects, because there will be many other effects happening in there that we cannot account for them today.
Yep.
So this is only the start of the journey, if you like.
Yeah. And Carl.
I guess the follow-up question from there is, if your surfaces do get infected, do they protect the bacteria from antibacterials, or do they come off easier? I think it's probably the key follow-on question.
Yeah.
Sorry, I couldn't really hear the question.
If the bacteria do get on that surface, because of the surface, are they easier to treat or remove? Is that right? Yeah.
It's an excellent question. In principle, in the surface with the curvatures, it should be much easier to clean that surface, to remove the bacteria from there. In terms of treatment, we don't know. That's something that we are looking to. I actually was this morning talking with Trevor about that, and that's something that we will be looking to do as a proper study in the future, but we don't know yet.
Yeah. And Andrew, the sinks seem like, I don't know, are MIME still doing their kind of critical unmet need, and then the engineers have a solution? Because you'd imagine the sink problem in hospitals is something that would be really great to have a group of academic engineers come. It's not just the piping, it's the physics of the water flow and splash, the angle of the faucet, and all these other things. So that would be a great
absolutely
MIME sort of approach. Yeah,
that would be fantastic. Our engineers in the hospital are really excited about it as a problem, and from the first time we spoke to them about it, they were coming up with suggestions, not just the phosphoric acid, the blue thunder, but also different
under sink.
dosing directly into the S band and things, and I guess they just don't have. There's a lot of things they don't have, but one of them is the time and the resources. So absolutely, collaborations would be terrific.
I'll just let you know that Andrew and I have been at Sync working groups for the last 5 years. We're it's sinks, and then we became ventilation experts through COVID, but now we're back on the sinks. So we're part engineers, but anyway, it's an ongoing problem. MIME grants, is it same process, unmet need, and the engineers?
As far as we know, it'll be the same sort of process, and blue thunder on the front of an application. I think that's all you need. That's great.
Absolutely. I think it looks like a great MIME application. All right, we might round out this, and I'm firstly put our hands together for speakers for this session. Really brilliant session to round out and think about prevention. So I think I'm going to hand over to Trevor now, who's going to round out the day. Thank you, speakers. That was fantastic. And I'm going to hand over to Trevor.
Yes, thank you. I'm not going to round out the day because I'm aware that everyone wants to rush down to the trains and the trams to get out of the city before the clock strikes 5. There's just two things that I wanted to say, and I'm sure I'm speaking on behalf of everyone here. So our Centre to Impact AMR is a bit of a maverick operation in a university. We don't do things according to protocol, at least not the standard and traditional protocols. We try and look for ways in which we can do things differently that would be helpful to the broadest part of our communities. And in this case, this is this community. The way in which we do that is to run working groups that look at big ideas that could be put into practice quickly to try and work out ways that we can do things better against the problem of AMR. Now, I'm saying that not as a spruik for the centre, but simply to say, where do our working groups come from? They come from brilliant people who care enough to come up with the idea of what do we need a working group on. And about a year ago, Nenad came up with the idea of the unmet clinical needs in AMR as a working group idea. This symposium is the culmination of the working group's best ideas, working with his team and with Kerry, the research manager in the centre, to conceptualise and bring together all of you for this symposium. And I am sure that I'm speaking on behalf of everyone, but it's definitely from me. This was fantastic. Today was really, really brilliant. I learned a lot. I met a lot of people. I was really rapt to see that I knew almost no one here. So, you know, there's the Centre to Impact AMR people that are here. Great, I know all of them. But there's a community here that's drawn from so many groups and so many organisations and so many discipline backgrounds that it was really thrilling. And I think that's why the symposium worked so well. So number one thing to say is thanks very much, heartfelt, to Nenad and to his group for putting on this symposium. And the second thing I wanted to say, and then you can applaud the organisers. The second thing I wanted to say is that we are now a community, right, of people who come from engineering, clinical, scientific backgrounds, from social sciences, people who've got all sorts of experiences to bring into this community. And I'm just letting you know that if this group would like to see a second iteration of this workshop, in a year's time, in 2 years' time, whenever we would decide it would be a good time to get together again, perhaps even bring in some more people by then, the centre would be really glad to try to organise such a thing and fund such a thing to make it run again. So biggest thing to say is thanks very much, Nenad.